proteins on its surface make it look like a king's crown. Hence, these spiky proteins, known as S proteins, act as anchors that allow the virus to invade a live target mammalian cell. The virus contains a single-strand RNA, which is the key component of the virus. Coronaviruses have the longest known viral RNA genomes, with a length of about 30,000 bases. This is a human cell, one of the main targets of the coronavirus. On the surface of the human cell, there are lots of surface proteins, one of which is called angiotensin-converting enzyme 2, or ACE2. Scientists have identified ACE2 as a protein that interacts with the S protein, leading to the entry of the virus into a target cell. After ACE2 recognizes the S protein, the virus is driven inside and begins to hijack the cell. First, the S protein and other surface proteins on the virus are degraded in the cytoplasm of the host cell, then assimilated.
A string of nucleotides which represent a portion of the viral RNA. This is viral RNA polymerase, which is a molecule that can make more viral RNA. Let's see how this works. These blue dots all represent nucleotides present in the cytoplasm. The viral RNA polymerase will use it to make more viral RNA.
This is the molecular structure of remdesivir, a drug developed by U.S. pharmaceutical company Gilead. It is a nucleotide analog which contains a structure very similar to adenine.
The virus co-opts the cell into producing copies. A key enzyme in this process is RNA polymerase, which synthesizes the nucleotide components that make up the RNA strand. Remdesivir appears to this enzyme as adenosine, one of the nucleotides. The RNA polymerase grabs remdesivir and attaches it to the chain. But after doing so, further nucleotides are unable to attach, thereby terminating the replication process. By preventing the completion of the replication process, remdesivir stops the pathogenesis for SARS coronavirus 2.